Hi everybody, welcome on Saturday afternoon. This is similar to what we did last year. This time it's a bit closer to the exam, so it's going to be less about trying to learn the content and more about how to answer the questions. And we have the advantage of having probably 20 to 25 QCAA markers on the team. So we have a pretty good insight into what the QCAA will be looking for. Does anyone know how they actually mark the exams and what the process is? You haven't been told that by the teacher. No. So the questions are marked by different people. So basically the senior teachers put their name in a hat, not figuratively speaking, and they then just draw out names and they are given a question to mark. What they're given is basically a bunch of keywords that they're looking for. They're not given very long to mark each question. Tens of thousands of students, and you've got a couple of weeks to mark all the questions. So what we will go through today is how to answer the questions. Obviously, there's going to be some content in there. How much to write, where to focus your time. Oh, here's this one. Come back in. Where to focus your time, that kind of thing. The exam will come in for 3 parts, but you'll do it I think it's 90-minute sessions. Is it still 90-minute sessions this year? I'm not teaching some this year, so.
Yes.
Yeah, so you get 2 90-minute exams. You'll get 20 multiple choice questions and 25 marks worth of short answer questions. Biology is a bit different because when you actually look at the paper 1 and paper 2, it's not like maths or chemistry where there's a significant difference. Sometimes they just put the medium and short answer or long answer all mixed in together in the 2 papers. The idea is you don't need to write, just because they give you a whole page, it does not mean you have to write a 1-page answer. If it's worth 4 marks, then you need to include 4 concepts. If it's worth 1 mark, you can get 1 mark by writing 1 line. Just because they've given you half a page or 5 lines does not mean you have to write in there. Some people will always run out of time, and sort of the way that I coach my students is that if you can't answer a multiple choice question in 60 seconds really confidently, move on. You should have those, you should be through the 20 multiple choice questions in 20 minutes. That doesn't mean you have to answer them. 20 minutes, 1 minute per question. If you cannot answer that question confidently in 1 minute, move on. Who's answered a multiple choice question, then got halfway through a short answer question, then realized that they actually remembered what the answer was because they were doing the short answer question, and vice versa. So that's my advice. 20 minutes. You've then got an hour and 10 minutes for the other 25 marks. So paper 1 is worth 45 marks in total. Pardon me, sorry. 20 multiple choice questions, get them out of the way in 20 minutes. That doesn't mean you have to answer all of them. But make a note in your head of what you didn't answer. And then as you're writing your short answer questions, you'll jog your memory to go back. Because if you've got 8 questions in paper 1, worth 25 marks. What's that? If you've got 70 minutes, good maths people, it's about what? 9 minutes per question. Lucy's having some connectivity issues. So you've then left yourself 9 minutes per question for the short answer. For that section anyway. So does anyone struggle with time management in an exam? You either finish in 5 minutes or you finish or you don't finish.
Yeah.
So use that perusal time to make a plan. You're not supposed to have a pen during perusal time, are you? Are they strict on that?
No, you're not allowed to use your pen.
Yeah. So make note mentally of the order that you're going to do the questions in. So pick the questions you can answer most confidently first. So the idea is that you go to those questions and you get on a roll. There's a lot of psychology behind doing exams. There's a lot of pitfalls. So get on a roll, get the confidence going, and just start writing. Get those easy ones that you know you can do. There's going to be easy ones like percentage abundance and what's a gene. They're always going to be questions like that. So just get in there and get them done. Get those easy multiple choice questions done. 20 minutes. You've got then got an hour and 10 minutes to answer 8 questions. 4 of those questions you'll be able to answer like that. They will just come out of your brain. This page. Whoa. All right, welcome page. So I think everybody's here. There's one person that's not here. So yeah, use that perusal time to make a plan. Don't start panicking, read the questions and freak out and put your head in your hands. I don't know anything. Make a plan. Bash out the multiple choice questions that you can answer in 20 minutes. If you can't answer all of them in 20 minutes, or at least the ones you know, move on to the short answer questions. Answer the ones you know you can answer. Bang, bang, get them done. If you've got a decent grasp of the content, you should be able to answer 4 of those questions comfortably. This is not the difficult part of the exam. You're not going to have 8-mark questions where you have to determine, you know, the likelihood of wrinkled green peas in F6. Like, that's not going to happen in the first part. How long is the gap between exams this year? How long do you have as a break? And what are there any restrictions on you in that break?
I think we have 45 minutes.
So 45 minutes. Who sits there and tries to study in 45 minutes? Anybody? Please don't. If you don't know something, you don't know the content, you are not going to learn it in 45 minutes. Use that gap, that break to just chill out. There is no point trying to learn anything new in that 45-minute gap. Some schools actually won't let you. They will physically stop you. Okay, so make a plan, get confidence going, get the brain juices flowing. Who gets exam anxiety and brain fog?
Yeah.
Yep, me big time. I was terrible at exams, probably still am, I haven't done one for years. Brain fog, anxiety can be cleared by stopping. Look out the window for 30 seconds. And if you suffer from exam anxiety and anxiety in general, having a plan of attack made in that 10 minutes will allow you to go, okay, well, I can just skip this question. The absolute worst thing you can do is sit there and continue freaking out. Turn the page, go to the next question. I've done a 10-week series on exam study and how to actually take exams, which I'll send you guys after this. And yeah, the absolute worst thing you can do is sit there and get frustrated. The second you get frustrated with the question, move on. There's another strategy, which is do an easy question, do a question you're not so confident with, then do another easy question you're confident with. So you're staggering them. And then you're not doing all the easy questions, then you hit 4 blockades. If you get frustrated with, if you do it that way, you do the easy question first, you know the answer, you're comfortable, it's fine. The second question is one that you're not so confident with, or it might be a more complicated answer, or it might be maths, because everybody hates maths. Although, like, I think like at least half of you have done methods exams, so you should be fine. And because then if you get stuck, you can then, okay, well, I'm going to move on to my next part of my plan, which is another easy question. And you break through that brain fog, and then you can go back to it, or you can go forwards to the next question. Hopefully you guys open the exam and you know all of it. That would be amazing. Who did the methods exam? Who found the calculator section harder than the non-calculator?
I did.
I did too.
Which bit was harder for you?
The calculator one.
Active, yeah.
Yeah, so probably there were some tricky questions, and I'd be interested to see the actual exam in a couple of weeks and mark it. Because so it's 50-50 split. Half the people thought the calculator exam was impossible, and the other ones found it really easy. So, but found the non-tech exam quite difficult. Okay, so what we're going to do today, now I've given you my spiel, my rant. I'll send you guys the exam prep seminar. It's probably only like the last 2 hours. Would be worth watching. Probably a little bit late now, but I'll send that out to you guys. So we're going to go through the multiple choice questions from last year as a bit of a warm up. And if you know the answer, I'm just basically going to give you guys 60 seconds. We're going to treat this like the exam. You've got 60 seconds. At the end of that 60 seconds, throw your answer in the chat. If there's a little bit of confusion, we'll go over the concept. If not, we'll go through. And then we'll look at the harder questions. We'll probably do 55 minutes, take a 5-minute break, and then do another hour. Everybody okay with that?
Yeah.
So multiple choice questions. What are they testing your knowledge of? Or what are they testing you on? Your ability to do multiple choice questions. I know that seems silly, but this will separate this is 20 marks, and you should comfortably get 15 marks. So we're going to go question 1. Again, start off pretty easy. This is a little, this is almost back to unit 1. Let's set a 60-second timer, just so you guys can get used to the pace that I think you should be going at in the exam. Obviously, you can't, they're not allowed allowed to even wear a watch, or they just banned all watches now. All right, 60 seconds for question 1. Once I say go, we'll throw the answers into the chat. Again, this is a little bit of unit 1 stuff. So we're going to practice pacing these kind of questions. 60 seconds goes by pretty quick. So this is part, actually I lie, it's kind of from this part of unit 1, part of unit 4, but this is part of the biotech unit. So when we're looking at DNA in prokaryotes, we are looking at DNA that's on plasmids in the cytosol. Prokaryotes don't have a nucleus. Only plants have a chloroplast, and the mitochondria is not where the DNA is stored. So if you're thinking this, it's biotechnology. They're sitting in the cytosol, in the free space in the cell, on plasmids. So I think everybody, so if you didn't get D, hopefully that makes more sense now. The way to remember prokaryotes and eukaryotes as in U, complex, nucleus, membrane-bound organelles. Prokaryotes are bacteria, simple, single-celled, no membrane-bound organelles, and their DNA is stored in the cytosol. The one thing they didn't have much of in this exam is biotech. I think that will be in this year's exam. I don't think they'll go 2 years without assessing a part of the curriculum. So at the end, we'll talk about biotech as well. Okay, question 2. We've got 60 seconds. Just try it before you put your answers in the chat, give it like 30 seconds or something because everybody can see the answers then. So I'll give a signal after like 45 seconds, everybody can put those in for the next few questions. So the overwhelming consensus is A, polygenic. Why? What does polygenic mean? All right, so polygenic is multiple alleles will determine the trait that is produced or the phenotype. Yeah, poly means many. So many genes produce one phenotype. So we get a very large level of variation. Yeah, so large level of variation with polygenic. All right, I'm gonna give everyone, again, this is one of those ones that you should know. You've got 30 seconds for this one. You should be able to get through some of these pretty quick. So what is the genome? If you've got the answer, go. Yeah. So the one thing they should have taught you about is the Human Genome Project. So that is looking at all of the DNA, every single base pair, whether it be coding or not. So the genome is all of the genetic material. Another answer could be all of the coding and non-coding DNA sequences. So that would be a way they would trip you up. All right. So most of you guys are getting this at like 30 seconds and less. So, all right, this one, a little bit of a data test type question. So you're using the key here to answer the question. Again, this one will be a 60-second question. What this also allows you to do, if you say, well, I've got 20 minutes for the multiple choice, you could leave this question and move on. Because if you get a load of others in, what's the word? That you can get done in 30 seconds or less, it allows you time to come back to these more complicated ones. But if you're on a roll and you go, I'm going to leave this one, jump to the next one and keep on that roll. Get to the end, then come back to any, have a look how much time you've got left. Oh, cool, I've got 5 minutes left to get 2 questions done. It allows you to spend a bit more time. You know, most of the time when we look at coaching for exams, it's not the content knowledge that is the biggest improvement, it's the ability to actually undertake the exam. Okay, anybody have any ideas with this one? Anyone that's done chemistry will be a slight advantage here. Okay, so overwhelming majority is D. So we're looking at this key. So we're looking at the biotic description, a small amount of low-lying moss. Okay. Is it moss and lichen dominated? No. Is the soil nutrient rich? Well, let's look at the reference ranges. No. Is it acidic? What pH do we need for acidic?
Less than 7.
Less than 7, yeah. So we've got 6.1. So again, barely even need any biology knowledge for that kind of question. Again, this is another one. where you don't really need to know anything about biology. All right, I'm going to give 30 seconds for this one. As long as you can read a graph, you can answer this kind of question. What you'll find is that over the course of the years, the questions might have changed, but the types of questions you'll encounter, like reading a graph, will not. So evolutionary radiation is a fancy word for number of marine families. So which had the greatest increase? Or division, that's how you pronounce that? Yeah. So you're just looking, so from 200 to 480, nothing else has had that kind of increase. Perfect. Again, a nice easy one that you can get on a roll, get that done and move on. Carrying capacity of an ecosystem. Think logically about this one. Yeah. Again, another question that everybody got in about 25 seconds. And this isn't questions that I've just made up to be easy to make you guys feel good. These are QCAA questions. So just remember that when you're going through. All right. I don't think I need to explain carrying capacity to everybody. You all got that one right. Some of you would have had an exam very similar to this or exactly the same as this for your mock exam. All right. If you know the answer, you can put it in the chat. C, yeah. What's K-selected species? And what's the other kind of selected species that we look for?
Are selected?
Yep, so we've got K-selected species, which are,
Low reproductive, high lifespan.
yep, so humans are selected species, which are your opposite, so rats or ants, whatever you want to use as an example. So as you're progressing towards the climax community, that's the peak, that's when you're hitting, you're about to hit the carrying capacity. So you'll have an increasing level of biomass because once it hits that climax community, it'll fluctuate up and down because the carrying capacity can be influenced by rainfall, sunlight, those kind of things. But as we're approaching the climax community, everything is increasing. Biomass just being the total amount of biological material available, plants, animals, whatever. Sweet. All right, again, you don't need any biology knowledge for this. It's some pretty basic maths. All right, what do we got? A, yes. So again, this is basic maths. You take the sunlight that came in, the heat energy from the sunlight, and then you take away everything else. So this links to those trophic levels, and we'll get into those in the other questions. Okay, this question here, myself and 2 other teachers have tried to contact QCAA because seemingly the answer that is correct is not listed. So how do we figure out the population growth rate here? Anyone, any ideas? So we will take the final population, which is 1100. We add the births, which is 290. We add the immigration, which is 182. And we minus. And then we add the deaths and the emigration, and we take that away from the total. So anybody able to get an answer for this one? How we find out the So I'll do it on here. So we have 1100. Can you all see me using the calculator on Google? So 1100 plus 290 plus 182, which is our immigration, gives us 1572. We take 1572 and we take away our deaths, which is 167. And we take away our emigration, which is 140. So we have 1265, which is our population at the end of 2011. So we've got 1100. So 1.15, take away the 1 and it gives us a 15%. So QCAA have not been able to give me the answer I was looking for. If we do 1100 divide 1265, it's 11,000, gives us 86.9. So don't worry about the answers there. Still have not been able to get an answer out of them if they made a mistake or not. But that's how we do it. Okay. Again, 60 seconds. We're looking at PCR here. So question 10. So, Alex, what did I say? Oh, no, I haven't used this for PCR, an acronym for PCR. So PCR is looking at infinitely small number of very particular DNA fragments. So the COVID test is a PCR test. So what actually happens is you get the shoved up through the back of your nose, feels like it's in your brain. What we then do is take that, put it into our PCR well. Some tests are as specific as 1 or 2 fragments of DNA, so they're very, very sensitive. We denature the DNA, so we split it into 2. We have what's called Taq polymerase that then goes in and amplifies the target DNA. You know, it does this, and every time this polymerase makes a new copy, another cycle of the DNA fragment we're looking for, it basically makes a brighter and brighter light, for lack of a better term. And once the machine registers that, okay, that sample is bright enough, it then becomes a positive test. If the target DNA isn't in there, nothing will happen. All right, question 11. Obviously, we're taking a bit longer than 20 minutes to do this. So we decide, yeah, Linnaean system, physical features, not the best. So again, this one's pretty easy. Yep, A binds with T, G binds with C. Perfect. So question 13, probably our trickiest one so far. So RNA polymerase. What does RNA polymerase do? And how is DNA helicasing? DNA helicase unzips the double bond. It breaks the weak hydrogen bond and it creates the replication fork. RNA polymerase comes along, forget where my camera is, comes along, attaches itself to the promoter and starts making the messenger RNA based off the template strand of the DNA. Okay, cladistics. If you don't know cladistics, please go and study it. So cladistics, existing population, 2 groups. You'll see that when we look at a clade. Question 15 again, another one which I queried and never got an answer for, so I'm not going to go through it. So question 16. Overwhelming B. Perfect. Species 2, not going to live down here. Species 2 has the competitive advantage. Now we're looking at the ecological niches that they would inhabit. All right, question 17 again. Another one that it's all in the question. Basically. If you know roughly what you're talking about, you should be able to answer this question. See, yeah. Again, we've got three chromosomes. Trisomy is an additional chromosome. Monosomy is you only have one. So it's not going to be those two. Trisomy 21 is Down syndrome. Klinefelter syndrome is a sex-linked disorder. All right, question 18. You guys are smashing through this. So everyone's saying C. Perfect. Dark colored butterflies and light colored trees, they're going to get eaten. The big A is going to reduce. Little a, because it's going to have more abundance within that population of the white butterflies because they're not getting eaten, it's going to increase. Very good. You guys are smashing it. Question 19. So page says, so yeah, the answer is B. Okay, again, that's a simple one of just looking at it by eye. Question 20, and our final multiple choice question. So micro and macro evolution covers pretty much all of our evolution types. So they got mutations, gene flow, and genetic drift. All right, are you guys confident that you could set that target of 20 minutes? And I think most of you could probably do it faster. So 20 minutes. If you're looking at a question and you don't immediately know the answer in your head, skip it. It will probably come back to you. That doesn't mean, I don't know, actually, I don't know what I was going to say, that never mind me. So 20 minutes, set yourself a goal, 60 seconds per question. You should get, judging on your responses to these, you should get 10 or 12 of those in 20, 30 seconds. So you're giving yourself as much time to anticipate that you're going to get frustrated, you're going to get brain block, you're going to get stuck on questions. And if you kind of put those delays into your plans intentionally, then you're not, if it does happen, you can just sit there and accept that it's happening. It's going to happen. Give yourself every opportunity to sit there. Okay, it's happening. All right, look out the window for 30 seconds. Do some breathing. Do some, 5 things I can smell. Bring yourself back. Okay, that was only 30 seconds, but I'm okay because I have a plan. And having that plan gives you control. Even if things go wrong, you've still got that plan. You still know how much time you've got. You've allocated time for all of your questions, and it's okay because you do have time up your sleeve. So take control of the situation. own it, accept that you're going to get frustrated and brain fog, especially those, you know, anxiety and I know there's a few of you that have things, you know, dyslexia, ADHD, you've all got things that make it difficult, but you know those things are going to make it difficult for you. So put them into the plan that you have for that section. All right, let's jump on to the small answer questions. That's good enough. Again, there's some of these that you should be able to do in 60 seconds as well, like this one. This question is answerable in 60 seconds. And it's 2 marks. The 2 marks are writing down the formula, getting the correct answer. Can you see question 21, just to clarify? Okay. So now we've got 8 questions. 8 questions, and we should have 70 minutes to answer. Well, let's plan for 5 minutes of brain crying and just tears of agony. That will probably happen. There'll be people in there. Then you've then got 65 minutes, 64 minutes, 8 questions, 8 minutes per question. This one should take you about 30 seconds. The next question, you've now got another 7.5 minutes up your sleeve. So to get the full marks, you would need to say yes, 25% abundance, but percentage abundance equals the number of species B over the number of total species times by 100. Do not just stick that into your calculator and write the answer every step of the way, just like you would in a methods exam. Do not give them an excuse to take away marks, your hard-earned marks. All right, that took a minute and a bit. So we've now got what, 62 minutes, 63 minutes to answer 7 questions. So now we're at almost 10 minutes per question. We look at the second question. Again, question that, if you know the content, if you know what a clade is, bang, 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 it should not take you long at all. Again, I'll give you guys 1 minute for question A. Yeah, so a group of organisms from a common ancestor, as well as any other descendants from that ancestor. Perfect. 45 seconds. So where's the example of a clade on there? So you would just circle around A, B, and 1, around them 1 there. That's an example. You could circle 6, 7, 5, 4, 3, 2. It doesn't really matter. Yep. So which of the nodes represents the most common ancestor for species E and G? Four, perfect. So which 2 species shared the most recent common ancestor? D and E, yeah. So closer to the top where the split is, the newer it is. Okay, so we had what, 70 minutes for 8 questions. We've just finished the first 2 in what, 3.5 minutes? You now got 65 minutes for 6 questions. A good strategy to use is to start in the mindset of adding an extra question. So pretending there's 9 questions, dividing up your time, and having 9 questions theoretically allows you that time to proofread at the end. So if we've got 70 minutes, we pretend we've got 9. So we've got about 7.5 minutes per question at the start. That leaves at the end all that extra time to go back through and make sure you've answered it the way you wanted. All right, smashing it. Again, question 23. And it's really important. We'll talk where you guys are going, but in exam 2, you've probably got 11 questions worth the same amount of marks, maybe 10 or 12. It really depends on how they write the exam. So getting those good habits now in this first exam will make your life a lot easier. And you can, so when you're looking at this, there's equal marks in exam 1 and exam 2, 45 marks each. So you can just about pass the exam in this section. So 35 out of 45 in this section, which is not unachievable, could put you, you just have to get a few questions right in the second part and you've got your pass mark for the exam. Okay, so R selected, high offspring, low parental care, low lifespan. K selected, like humans, are the opposite. Again, that's another 1.5 minute question. Okay, question 24. Getting a little bit into the nitty-gritty now, but you've just gained 5 minutes per question in time by getting those questions that you know, bang, 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 out of the way, easy. We'll have a little 5-minute break after this question. Sorry. Yeah. So the genotype is the combination of alleles. So alleles, we know, are the genes that encode for the phenotype. The genotype is just the combination of alleles that an organism has. Okay, so question B, 24B. These are the kind of questions they're going to write to try and trick you, right? So most people will look at this question and start answering, what is a mutation and how does it occur? Is that what, did anybody think like that? So what you're actually being asked here is, how is the mutation inherited? Nothing to do with what the mutation is or how that mutation came about, just how it's inherited by the next generation. Again, getting a little bit tricky now. So what this strategy allows you to do is to spend more time on questions like this. So what we're looking at is say women do not make new eggs once they're produced. A certain number are produced and released over a lifetime. Say this woman smokes and is accruing mutation in her eggs. So what this gives us is a new allele or a variation of a gene that was there before. So Paige, you are correct in that some of these mutations may stop the organism from reproducing, but what the question is asking you is what happens to that gene? So you've got sperm and egg cell. The egg has a mutation, sperm fertilizes the egg, we have a new embryonic cell. That embryonic cell is going to carry the mutation from the egg because it was present there at fertilization. This will then affect the genotype of the offspring. Yep, Priscilla, probably as good an answer as you can get. So the 2 marks they're looking for here would be passed on to offspring through fertilization because the mutation was there in one of the gamete cells, the sperm or the egg cell. All right, let's take 5. Have a coffee, get some water, and then we'll reconvene. I'll just pause the recording. So who took biology because I thought it was the easy science? Or the school told them it was the easy science? Yeah, everybody. It's not a difficult subject, but it's generally taught terribly. And you just have this just a never-ending stream of content thrown at you. And B, Paige, that was silly. It's not, it depends on how your brain works. So biology is not mathsy, it's not massively difficult. I love how this like HD webcam shows the bags and all that. It's really good. I just now completely forgot my train of thought. But the idea is that biology should be easy. It's not, content is not overly difficult, but there's just so much of it, so much to it. Chemistry, I think I went through the 3 and 4 chemistry curriculum last year in 3.5 hours in one of these sessions, the entire thing, 3.5 hours. I think I'd struggle to do that in 7 or 8 hours for biology, and I'd be completely brain dead by the end. All right. Again, nice and simple one. This is the kind of thing you're going to expect in this. This is simple, familiar, and maybe some simple, unfamiliar as well. Thing with biology, there's not a lot that should be unfamiliar. It's just the way the questions are asked. So here we're looking at the inflow of energy versus the outflow of energy at each trophic level. If it makes you feel any better, I have 2 assignments due on Thursday. So I'll be spending my week studying with you guys. All right, what have we got for the net productivity of the producers? So producers, we have 52 minus 16 minus 8, because the plants are respiring and losing 16 megajoules per meter squared per year. And through decomposition, they're also losing 8. The herbivores, again, we start with 28. We take 20 and 5. So Alex, I have the QCAA marking guide that they use to mark the exam.
Yeah, I know. It was very, very funny.
Yeah, it happens
Like-
quite a bit when teachers tell my students that they're wrong, and then I say, well,
Yeah, so yeah, I don't actually know what to do because apparently all the bio teachers got together and had a big meeting and were like, no, you only do respiration and stuff. You don't minus the decomposition off. That's wrong. So everyone who minused all 3 got it all wrong. And I was one of those people.
yeah. So again, QCAA marking standard.
Yeah, they even admitted it. They were like, so the QCAA says do both of them, but we know we are correct, so only do this one.
Yeah, but QCAA marked the exam, don't they?
Yeah.
Not your teachers. So I'll prove it to you, and I'm not lying to you. Look. Down here. My advice is listen to QCAA, not your teacher.
That's what I was planning.
Yeah. Yeah. There's a bit of a divide between QCAA and reality, unfortunately. Okay. So what does the cognitive verb contrast mean? You don't have to explain it. I'm not asking you to explain. Yeah, just say this one was this and this one was that. So you can say the numbers. Or you can work out a percentage. Literally state the numbers. So there was the difference is 4 or 16 and 20 in respiration, and 8 and 5. It's literally just stating exactly what you calculate. You could also compare the percentage loss at each level. So 46% for producers, 89% for herbivores. And then you just state the percentage loss is higher for herbivores. Very simple. So microevolution, what are we going? Yep, Ben, you probably need a little bit more detail than that. And it's not really the time length that is the differentiator. So So it's nothing really to do with the time in this answer. So Priscilla and Ben, if we combine your answers together, small scale changes in allele frequency within a species or a population of that species. The other part is we're looking, it has to be the same taxonomic group as the ancestor. So this new evolution. Again, it's only 1 mark. It is, you need 1 sentence to answer that. $27, and then I'll do a few more of these, and then we'll jump over to so I'm going to give a little bit more time for this. Do you see what I mean about making a plan? Because most of these questions, you could have answered in 2 minutes. Yep, so we've got coevolution, so they're evolving together. So how have they evolved these characteristics? Again, you don't really even need any biology knowledge, you just need to be able to read the question. Yeah. So to break it down to 3 parts, you need these key terms. So crabs, they get the stronger claw randomly. They can now crush the snails with the weak shells. The first paragraph, you state that, or the first sentence is maybe a paragraph. You're stating that the crabs with stronger claws place a selection pressure on the snails with the weak shells. So that's 1 mark. So only the crabs with the strong shells will survive, and those snails, through selection pressure, will evolve to only have the strong shells. So as this is happening, the crabs with the weak snappers are going to die off because they can no longer break the snail's strong shell. So we have evolution occurring in both populations in response to each other. So, and remember with evolution, it all comes down to the alleles and the genes. So what we will see in both populations is a higher allele frequency for strong shells, strong claws. So that's your 3 marks there. Selection pressure, they're evolving together, and the allele frequency is changing to be more of the strong shell and the strong claws. Does that make sense? Okay, cool. So, time. We've got 2 more questions. So 4 questions. Sorry, 4 marks. We have primary and secondary succession. 1 mark is 1 difference between them. The other mark is 2 differences. Sure, all right. The other 2 examples are, the other 2 marks are an example of each. Doing all. So if you want to have a look in the chat for Ben's answer there, that would be full marks. Priscilla. Yep. Paige. Perfect. Have a look in the chat for the answer. I can't really make any of those any better. So primary succession, volcano. The only other thing that you could look at is talking about the keyword here, pioneer community. So you need some kind of hardy moss and lichen and your CAM photosynthesis succulent plants just come in and to fertilize the soil for you. That's the only thing I could really add to those answers. All right. So even with me talking, and probably didn't start till it's taken us about an hour to do that exam. So took us just over an hour to answer all the multiple choice questions and the short response questions. So if this was your exam, you'd now have half an hour to make sure everything's perfect. Guess what you don't lose marks for? Writing too much. Write the bare minimum that's required in the that you think is required as you're going through. Then you can always come back and as you're doing the exam, things will jog your memory. And remember that you've got the extra paper, right? Just have it there. Every time you think of something, oh, okay, I remember now, write it down. Do not try and remember it. You can tell I've been ranting about exams. My voice gets like this. All right, who's ready for the big questions? They're not, this is biology, right? They're not going to be hardcore examinations. Okay. So this section, we have 11 questions to answer in 90 minutes. So we're gonna pretend that we've got 12 questions in 90 minutes. So you're gonna keep track, and we're gonna have 7 minutes per question. That then should give us, if it takes you 7 minutes to answer each question, we've then got 10 minutes at the end where you can go back and proofread. All right. Voice is going. What is a symbiotic relationship between two species? Again, 2 marks, you need to make 2 points. I have too many things open. You see the questions now? Okay. I was just trying to help. You see the question now, not the answers? I don't even have the answers open. How can you see the answers? Okay. I like how you waited like, a good minute before you said anything, just to make sure you got them written down. Okay. It's not a Zoom. Yes, this is, can you see the symbiotic question? Okay, this is paper 2. It wouldn't be a Zoom session with me if I didn't. So Paige, yeah, that was 28 questions for paper 1. The 20 multiple choice and the 8 short answer. Most of these aren't going to be that long answer either for paper 2. They're just a little bit more complex. So, Priscilla, your answer is pretty good. The only thing I would change is just changing the way you've written the long-term relationship. Yeah, actually, that's fine. You have to have a real trick of a marker to not give you the full answer for that. So, interaction in which the species live together in a long-term relationship. Pretty simple. What is mutualism? Again, hopefully you guys can see that. Yep, Alex, perfect. What's an example? Yep, so we got 2 species that both benefit. Clownfish and anemone. Zooxanthellae and coral. Yes, Priscilla. What have we got? Zooxanthellae. I'm not good at English. Is the name of the algae. Okay, cool. Yes, Priscilla, that's the one. Zooxanthellae or Zooxanthellae, depending on the species. Okay. Getting a little bit more complicated now. So we're going to have 2 minutes of thinking. So the thing about the QCAA marking standards are the way that most courses are designed now is under what's called universal design for learning, which means that you can't penalize people for displaying information in a slightly different way. So if you actually look at the marking guide, underneath everything it says, or other suitable response consistent with a reasonable understanding. That is on the end of every question where they talk about the marking. So even if you are not, even if you're just racking your brain and you can't think of, just write something. Reword the question even, just put something down. Because if you can show understanding, even if it's not perfectly worded or it doesn't make any sense, you can still get a mark. So what's the principle though? So yes, competitive exclusion. Should you just write that? Really. Okay. So we have 2 species here that, based on the answers, or based on the data. Sorry, I'm just getting over the dab. We've got 2 species that occupy the same ecological niche. And that's proven by the fact that together, I mean, sorry, my voice is gone. Separately, species A and B live happily. Species A and species B cannot live together. So for part B, actually I'm going to give you another minute for part B. So if there's 3 marks, what does that tell you we need to do? We need to have 3 points in our answer. Yep, so page, species A has the competitive advantage over species B. So when you're looking at the separately, test tube 1 and 2, you can say that species A likely has a higher reproductive rate, or it's more efficient at using the resources. So it will then outcompete species B in the same test tube. Why are the numbers lower in species A in the third test tube than the first one? Because B would have used some of the resources. So the 3 marks here, yeah, there was competition in tube 3. Species A and B competed for the same resource. Species A outcompeted species B. The third part will be species A's population was affected by the competition in tube 3. Very good page. Right. Give everyone else a second to catch up. All right. Question 3. So we spoke about helicase. So what does DNA helicase do? Yep. Again, the reason I'm getting you guys to put it into the chat is because you can use that to formulate your own answer or improve your own answer. So helicase breaks, so in order to get the 4 marks here, helicase breaks the weak hydrogen bonds between the bases. It unzips the DNA and it exposes the bases so that polymerase can run along. DNA polymerase adds the complementary nucleotides to the exposed bases. Because what we're doing here is we're creating an identical copy. Now, there is one more thing that DNA polymerase does. There's one more function. Yeah, it proofreads the newly synthesized strand and checks it for mutations. So again, there's 4 marks there, but you could effectively write 50 words to get that. So being concise in your answers, which you guys are doing in the chat like brilliantly. Yeah, you guys are doing in the chat brilliantly. All right. You guys given a forming book for biology this year? I didn't do grade 11 bio this year. I'm just saying. No, I don't think so either. A lot of the times you'll get given the formula. And it's just a matter of, so what's the epsilon mean? Sum of. So I'm trying to find my whiteboard mark misplaced. The big E looking thing, yeah, epsilon. All right, I'm back. So you can see here, 1 minus what's the second number? Although you guys can't see that screen, so I don't know why I'm asking. 14. The final one is 8. 9 by 99, which is the total. What does that give us? Anyone's got their calculator there? .37. Perfect. Who is feeling slightly less worried about their biology? No. I don't think any of you should be worried. Ben, I think you're just in panic mode. Don't think much is going to make you feel better about your exam. All right, does that make sense? All right. I'm just looking at time. Let me get through these questions. So, proteins, can everyone see question 4? Sweet. I'm not sharing the answers again. You're not just telling me that so you can see the answer. Okay. So again, you are not expected to provide that much information. So I'm going to give 2 minutes for this. Ben. The only thing you didn't talk about there, Ben, was transfer RNA. So transfer RNA brings the amino acids. Page, tRNA and mRNA are both in translation. Might even draw one of my famous diagrams. They're not famous, but terrible. Okay, so I want you to write down 4 very simple steps. DNA is copied. mRNA is produced. mRNA goes to the ribosome. tRNA adds amino acids to form the protein chain. And whilst you guys are writing that down, I'm going to draw a beautiful diagram. If I have something to rub the board off with. I swear I had it with me. I think I left it on the coffee table while I was making it. Had every intention of being with. So, transfer RNA adds amino acids to the growing protein chain. So we have our 2 amino acids. Transcription starts when helicase doesn't stick. So helicase unwinds the DNA. Exposing the strand. This is our gene of interest. So we copy the DNA. We make a copy of it. RNA polymerase binds to the promoter. RIP my red pen. So, as it's running along the DNA strand, it's producing a copy of itself in the form of an mRNA molecule or messenger RNA molecule. So RNA polymerase binds to the promoter. It's running along the gene of interest and creating an mRNA molecule based on the codons that are present. Do you remember in DNA we have GATC? What bases do we have in DNA, in RNA? So GATC for DNA. GAUC for RNA. So here we've got our RNA molecule. This is our messenger RNA. Let's make it a linear, not a circular. So this is where I think it's actually taught wrong. The ribosomes sit in the endoplasmic reticulum usually, right? So the messenger RNA will usually go into the rough endoplasmic reticulum. And there's two subunits of, actually there's three subunits, but we'll just do two of the ribosome. So it's not like the ribosomes are just like fixed in place in the cell. They're in subunits, they're broken up, and we get the large subunit on the top, and then we get the small subunit. Well, there's not much of a difference. So this is our ribosome. Over here, we have tRNA. So transfer RNA. What it does is it brings along the amino acids. And it's got anticodons that bind to the code for methionine, whatever. And as the mRNA goes through the ribosome this way, the protein chain is growing as well. So 4 marks. Concentrate on the 3. mRNA molecule is produced by RNA polymerase. mRNA is transported to the ribosomes, and the 2 subunits of the ribosome attach to that messenger RNA. Once this happens, the transfer RNA bring the amino acids to the ribosome. They then attach the right amino acid into the right codon spot. And that's translation. Clear as mud. So Paige, the ribosomes are made up of subunits, and they won't start producing the protein until all the subunits are in place. Again, you don't need to know a huge amount about the subunits, but you'd impress a QCAA marker if you do that. So Ben, I'm not sure what carries the mRNA to the ribosome. I think it's just a chemotaxis. The transfer RNA brings the amino acids that correspond to the code in the mRNA, and they start to build the protein chain in the ribosome. All right. I think we've got time for one more question. I think my brain has one more question. All right, can everyone see question 6? Sweet. I'm paranoid. I think this will be the last one. I don't know if I have much more of a brain to take. Yeah, food is probably the easiest one. Other things I wrote down: water, space, shelter. So how could the carrying capacity be negatively or positively influenced? Yep, increase in predators. What else? It's on an island, so space, land clearing. So Alex, this could be anything. Doesn't have to be humans. It could be natural disaster, like Ben just said. An introduced competitor, yep. So for this, to get the 2 marks, you would need to provide a cause and a consequence. So new development on the island, taking away space for nest. Getting rid of the humans, Alex. Increasing food supply, increasing water availability. If you're wondering how we could positively affect any environment, getting rid of humans is usually the first. If in doubt, just get rid of the humans. All right, I'm going to skip the next one and go to question 8. So I'm going to give you guys 60 seconds to think about this. J curve, very good. So exponential J curve. Why is it a J curve? Perfect, Alex. Do not think too hard. All right, what's the time? I think we'll leave it there for today. Yes, rapidly declining and rapidly increasing would be the most correct answer there. So who is feeling better about the exam? Who's going to go into the exam? And you can apply that kind of thinking to any of your exams that you've got coming up. Split it into manageable, workable chunks, and allow yourself time to freak out. Allow yourself time to get stumped. Allow yourself time to get brain fog. Allow yourself time to stop for a minute or 2 or 5 minutes and just stare out the window. Just creepily stare at somebody else in the room, across the room, and see how long it takes them to catch that you're staring at them. And if you get that sort of, yeah, wink at the QCAA monitor, just make weird faces at them. But yeah, if you people freak out when you don't allow yourself time to freak out. And that's the biggest thing. Give yourself time to freak out, and you won't and then you start freaking out because you wasted time, and then you freak out about, it's a never-ending cycle. So give yourself the time to freak out. You guys will be fine. Obviously these questions aren't going to be the questions on the exam. But you guys showed a pretty good mastery of the topic, except when I showed you the answers and you had all the answers. So then, if you have anxiety in an exam, plan for it. You know it's going to happen. Accept that it's going to happen and plan for it and be okay with it happening. Thanks, guys. It's been a pleasure. I think I might see some of you tomorrow for maths and then next week for chemistry. Exactly. I'll see some of you guys at chem. Be fun. Chem is my favorite subject. And I will have, I think, so I don't know if you guys can see that back wall there in the tutoring center. It's a giant blackboard. So chem, what I'm probably going to do is just set up this webcam facing the blackboard, and we'll just get weird with the blackboard and some chalk, and we'll get through it. All right, guys, I'll see some of you tomorrow and some of you next week. Some of you I'll see with tutoring. See ya.
Bye.
See ya. Wow. Oh, she said thank you.